Okay, it's 2:05, so I think it's probably about time to kick off. Welcome everyone to the third UK Hydrogels Network online seminar. Just a little bit of housekeeping. If you've missed either of the first two, the recordings are available on the website and the link is on the screen share at the minute. But today we're going to be hearing from Laura Miller, who is a lecturer in the Maths and Statistics Department at the University of Strathclyde. Before that, she was an EPSRC Postdoctoral Prize Fellow at the University of Glasgow, and where she did her PhD as well on the fusion and mechanics of multiscale biological tissues with Raimondo Penta. Am I correct in all of that, or is Google correct in all of that information?
Yes, it's all right.
Yeah, and we're really excited to hear what she has to speak to us today about some perhaps cardiac tissue related applications of poroelasticity. So I'm going to stop screen sharing and hand over to Laura, and yeah, we'll hear what she has to say.
Okay, so hopefully you can all see my screen now. I'll just make it full screen. Oh. Sorry. Get there. Doesn't want to go full screen. Mm.
I think there's an option under View in that menu you've just had up.
All right.
I'm not
Oops.
sure, but the menu at the top.
Usually the Control L works, but it's not working in this case. This might be.
They've changed it all.
Okay,
There
hopefully.
we go. Yeah, that works.
Okay, that says now. So thank you very much to Joe for this very kind invite, and I'm delighted to present to you this work this afternoon. So today I'm going to be talking about the micromechanical analysis of the effective stiffness of poroelastic composites, and then the application of this model to myocardial infarction. So this talk combines 3 different papers, which were all part of my PhD thesis. So it should be an overview of a variety of things I've done in the last few years. And this is joint work with Raimondo Penta, who was my PhD supervisor. So I'll just start with a brief introduction to porous media, although I'm sure many of you here are very familiar with this already, so I won't dwell on it too much. So here I have the sort of classical image of porous media, just some sort of elastic matrix with a variety of holes of different sizes where we can have fluid flowing. And this elastic matrix can be one kind of constituent itself that it's formed of, or can be formed from many different constituents at different scales of resolution. So these type of materials are usually modelled using Biot's standard poroelasticity. And this was a framework that was developed experimentally, and it was a macro scale system of governing equations. And it was developed initially for sort of soils and rocks. And then in the 1980s, the this model was again derived rigorously, this time via homogenization techniques, from looking at the pore scale microstructure of such materials and upscaling to actually get a similar system of macro scale governing equations where you could match the coefficients with the already experimentally obtained ones of Biot. And this was done by Burridge and Keller. So nowadays, porous media are used in many applications. Still, models of rocks and soils are important, but more recently there have been many applications in biological tissues, in artificial constructs and biomimetic materials. So I'm going to focus in this talk on biological tissues and a variety of different examples where they can be applied. So I've included this image here of human bone, where by zooming in, we see that underneath, this is a porous matrix, very similar to this one I show up here, where the solid part is a network of sort of collagen and minerals, and there's a fluid flowing in those pores. And what that does is actually create the macro scale stiffness of bone tissue. So these are the sort of things that my modelling takes into account. It wants to look at various underlying microstructures, create sort of equations that can describe the underlying nature of the material, and upscale them into systems of partial differential equations that can describe complete tissue systems, while still retaining an understanding of why the material has the properties based on what's happening underneath at that pore scale or micro scale. So the first sort of mathematical model that I'm going to present was actually the first model I wrote down as a PhD student, and this was my first paper published. So I always like to include this one in talks because it's the first sort of piece of work I was very proud of. So this is a paper on deriving the effective balance equations for poroelastic composites. So I should first define what I mean as a poroelastic composite. So that is a material such as this piece of human biological tissue that I have a sketch of here, where we have some sort of solid phase, some sort of second solid phase, and pores of fluid. So that means that I have been able to take a small section of this tissue, and I want to choose something that I feel is repeating over and over again and building up using lots of those small blocks to become the whole tissue itself. So I've chosen here a solid ECM or extracellular matrix, a blue solid elastic fibre here, and this white space here is a fluid-filled pore. And this is meant to be basically a copy of what we're seeing in this interstitial matrix tissue. So two solid phases, both elastic, and this empty sort of fluid-filled pore space here. So what I say is that this is a 2D cross section, so it's actually a cube with a cylindrical fibre and three interconnected cylinders to form a porous connected structure between all the cubes. And I say that this small unit is my periodic cell, and I associate with that a length scale d. And I say that if I put lots and lots of these small cubes d together, eventually I would create my full macro scale tissue, which has a length scale L. So that L is usually the length associated with the complete organ. So things like the bone, skin, the heart, whatever material you're actually trying to model. So this length scale is vastly different. So it takes many, many, many of these cubes to actually make up this whole material. So now to actually look at the modelling approach. So as I mentioned before, we take a structure that we think is representative of what we can see at a microscale level of resolution in our tissue. And we have to set up an equation that can describe each of the domains and the interactions between them. And what this does is it produces a fluid structure interaction problem, in this case, that we can then upscale to get a system of partial differential equations that will describe my entire tissue. So here I have three different domains. Omega 1 is my solid elastic fibre, and omega 2 is my solid matrix. So let's put down some equations for those solid domains first. So I have a balance of my two stresses, where T1 and T2 are just the stress tensors in each of those compartments, and they are given here by C1 and C2 respectively, which are the fourth rank elasticity tensors of each material phase. And these are the symmetric parts of our symmetric part of the gradient operator applied to the elastic displacement U1 in the fibre, or indeed the elastic displacement U2, which is in our solid matrix. So these are our two solid stresses. Then we have our fluid domain, omega F, which I assume to be a Newtonian incompressible fluid. So I'll have my balance of fluid stress tensor. My fluid is Newtonian, so my P is my pressure, my mu is my viscosity, and again, this is the symmetric part of the gradient of our velocity tensor V. And since I have that it's incompressible, I need this incompressibility constraint here. So these are the equations in each of the domains. And then I require some interface conditions to close the problem. So in this case, I'm going to have three different interfaces that I can describe. So the interface gamma 1 is this one that, if you can see it, is in green here, and that is between my fibre and the fluid-filled pore. And on that gamma 1, I have the continuity of the velocity with the time derivative of our solid displacement, or indeed just our solid velocity. And I have my continuity of stress tensor, so that's the continuity of the fluid stress with the solid stress in that compartment, and that's on gamma 1. I have the exact same set of conditions, but using different tensors on gamma 2, which is this interface around here between the ECM and the pore fluid. And on this gamma 3, which is going to be a solid-solid interface, because this is ECM and fibre, I have my continuity of elastic displacements and my continuity of just two solid stresses in this case. So that is all the equations that I need to set up that can completely describe this periodic cell and all the different elastic phases and interactions that are happening at that scale. So then what I need to do is exploit some sort of upscaling technique to reach a system of PDEs. So to do that, I am going to be using the asymptotic homogenization technique. So what I have done here is just resketch what I had in the previous slide. And you'll notice again, I have got these length scales D and L. The first thing I have to do if I'm going to apply the asymptotic homogenization technique is ensure that my scale separation parameter epsilon, which is a ratio of this length D to this length L, is much less than 1. This has to be the case to ensure that there's a sharp enough separation between my two scales to allow the upscaling to work correctly. So in this case, we are talking something that is on the scale of maybe 100 microns to the scale of something that might be 10 to 12 centimetres. So we're going to have an epsilon that's of the order 10 to the minus 4. So it's going to be very small. So that case is satisfied. So we can move forward with the technique. So then, using this technique, we associate a variable with each of our length scales. So I associate the variable x with my macroscale domain and macroscale material, and I associate the variable y with the microscale. I then assume that every field, so these are things such as the displacements, velocities, pressures that we saw in the previous equations, is a function of both of these variables. And each field is going to be y-periodic. This means that each cell is repeating over and over in our domain to create this overall material. And then what we need to do is perform a multiple scales expansion in power series of epsilon for each field in the fluid structure interaction problem. So I write every single field just as a power series in epsilon, so just as a sum of powers of epsilon. And then what I can do is also transform the gradient operator using the chain rule so that it is also a gradient operator of both variables x and y. And then I have all my equations written out in that form where they're very much expanded into these different scales, different operators that are all power series. And then I can equate the coefficients of different powers of my epsilon. So from powers of epsilon 0, 1, 2, and so on. And you keep equating powers of epsilon and creating closed problems until you close the overall system of PDEs in the end. So from these systems, from these equated coefficients, we form microscale cell problems that are going to allow us to determine the model coefficients. And these microscale cell problems are actually what's going to be remembering the underlying microstructure that we are seeing here once we're back in our full tissue, where we no longer see that there are different subphases or different pore structures or geometries or elastic properties happening at this microscale level. So that was a very, very brief overview of the technique. For the sake of time, I won't present the steps of the technique here. But for anyone that's interested, the paper is referenced on various of the slides throughout, such as at the bottom here. So we then just want to present our final macro scale model, which is a system of PDEs, as I mentioned, and we have our first equation is for the Darcy's flow that we find for the fluid flowing in the pores. So this W, lowercase w, we find here is just our relative fluid solid velocity that we have defined to make determining the equations a little bit easier. And we have this pink coefficient here, which is our effective hydraulic conductivity tensor. So that is remembering the underlying pore geometries that are at the microscale, even though we're now in a macroscale PDE. And that W is to be obtained via solving some of those microscale cell problems that I mentioned. And that is how it's able to remember the pore geometries and vessel geometries that we're going to be seeing in the underlying material. We then have a balance equation and our constitutive law. So our balance is just this balance of our effective stress that we've called it here. And this comes in two parts. So we have this tensor, fourth rank tensor here that I've highlighted in blue, which is our effective elasticity tensor for our material. And that is our what is encoding the fact that we have a difference in the elastic properties at different points in the microstructure. So that is again to be computed by solving some of the microscale cell problems. And it's remembering that we have our ECM and we have our fibre that can have different elastic moduli, so that they can have different Young's modulus, they can have different volumes, the different geometries. That is all being remembered in that tensor. And then we have this red tensor here, which is our Biot's tensor of coefficients, and it's relating the pore pressure to the changes in the solid mechanics that we're seeing. And our final equation is our conservation of mass equation. So this is relating the changes in fluid volume to changes in the strains. And we have, again, this orange coefficient. We can prove properties to show that it is equal to this red tensor here. So this, again, is our Biot's tensor of coefficients. And having this equality here actually simplifies the problem numerically when you want to compute the results of this model, because the cell problems that need to be solved to equate this tensor are different from the ones required to solve this one. But since those two coefficients are equal, we only have to solve the one set of cell problems rather than the two different kinds. So that is a very useful property to prove about our model. And this blue coefficient that I've highlighted is our Biot's modulus, and we can prove that it is a positive coefficient, and it relates a ratio of changes in pore pressure to changes in the fluid and solid volumes that we see in our material. So that is the model, and that was the new model that we came up with. Basically, the difference in this one versus other things that were already available in the literature was that this porous material can account for various different elastic constituents at the pore scale and the pores. Whereas in standard poroelasticity, you just have the porous matrix, which is assumed to be homogeneous, and the pores. So we wanted then, after we developed this, to understand if you really were seeing a vast difference when you used our model versus what was already available in the literature. So that's what I'm going to illustrate now. So I want to spend a few minutes to talk through this comparison that we come up with. So in the case of our new model, we have two elastic phases. In this case, we have this elastic fibre in the middle and our elastic ECM, and I have four just vertical cylindrical pores. The reason that we chose this geometry was so that, in fact, we could cut the plane and do 2D simulations to make things a little bit quicker numerically, although it's very, very possible to do the 3D simulations, was just a choice for what I presented here in this talk. So this is exactly what we wanted our new model to capture. Something where when you look at it under the microscope, you can see minimum two distinct elastic phases and pores all at the same level. And that was new. Nobody had ever tried to propose a model where you could see the three things all at the one scale before. But what was there in the literature was models for standard composites where you have two solid phases and that's what you see. Or you have just those typical poroelastic materials that I mentioned, where our matrix is homogeneous and we have the pores. We wanted to come up with the most sort of comparable way that we could to see what the difference was by having them all at the same scale versus having them not at the same scale. So we decided that we could create a composite where the matrix part underlying was porous. So this is a poroelastic composite. This is a composite with a porous underlying matrix. And if, when we did our simulations, we found that we were getting vastly different results between these two setups, we could be confident in the fact that it was worthwhile to have developed such a model, because if the tissue is showing the three phases at the same scale, you should be able to account for them all at the one scale, rather than doing this two-step process to try to best approximate what you're actually seeing. And spoiler alert, the model was worthwhile developing. So that was very good news. So here, just to explain a little bit about what we need to compute, I've got my very simple 2D cross sections of my material. So as I mentioned here, in our material, we have solid matrix, solid inclusion, and cylindrical pores. And this was our effective elasticity tensor that we needed to compute. So that was going to be reasonably straightforward simulations to carry out. We can compute this directly by solving cell problems that we developed in our model derivation. Here, as I mentioned, was the best possible case we could come up with using what was in the literature. And that means we had a two-step process. So what we had to first of all do was solve a just typical porous matrix problem and use that then as an input parameter for our composite problem. So this means that we had two effective elasticity tensors to compute here. We computed this porous matrix effective elasticity tensor, used it as an input, and then obtained this standard poroelastic approach effective elasticity tensor. So what we want to do now is compare the output from both of these two tensors here. And the full analysis of this is available in this paper that I've mentioned at the bottom here. Okay, so just to, again, put these tensors a little bit bigger so you can see them more clearly. So this is our two fourth rank effective elasticity tensors. This first one from my model, this second one from the next best case that we could come up with. So these are going to have 6 independent entries due to the geometries we chose. So the geometries, as I showed you on the last slide, were cubes with various cylinders. So that was why we just have the 6 independent values. And I want to calculate my Young's moduli and shears for both of these scenarios. So again, the fact that we have the 6 independent entries and the geometries that we have are the reason why I'm only mentioning Young's moduli E1 and E3 and shears C44 and C66. So these had to be obtained by doing an inversion of a elastic compliance tensor and manipulating various tensors, but we got our new expressions for our Young's moduli. And I'm using this theta subscript just to say that these are the components coming from the elasticity tensor for one model and the other model. So we've got two cases. And okay, so now to present some of the results that are coming from this. So here is a comparison between the two model cases for the Young's modulus E1 and E3. So E1 is the modulus when we are sort of pulling on the side of the cube where the cylinders are running vertically in this direction like this, and we're pulling this way. And the E3 is pulling in the direction that the fibres and channels are elongating. So that's what we're comparing here. And we can see that using our new model, which is this blue line, we get lower results for both of them than the standard poroelastic approach. So we see that we just did a test case where we were increasing the porosity because we wanted to understand what role the porosity was having on the stiffness that we could capture with both models. So we are seeing that the effect of the porosity is much more prominent when it's considered at the same scale as the other elastic phases. Now, this is probably what we would have expected, but that's actually reinforcing the necessity of our model, because with what was already in the literature, you can't have two solids and a porosity at the same scale. You can either have a composite with two solids, or you can have a homogeneous solid and the pores. So that already, before we even really were doing too much of an understanding of what this actual absolute difference was already reassuring, because you're seeing more of an effect already. And if that's what you've got in your material, that's what you want to be able to find. So then what I did was actually plotted the absolute discrepancy between the two models. So for our E1 Young's modulus, already when we're at about 5% porosity, which is reasonably small, we see that there is a 5% difference already between the two setups. And this just increases all the way up to 22% difference at 30% porosity, which I think is quite a large and distinctive difference to have between two models when you're basically trying to describe the same thing with the two models. You have a sample of tissue that has various elastic constituents and a fluid compartment, and you want to describe it. You have two choices of models. You shouldn't see as much discrepancy as that. So that therefore means that the comparative case that we feel was the best possible comparison wasn't really working for those types of materials. And similarly, again, with E3, we were seeing maybe a slightly less discrepancy, but still incredibly significant. At 10% porosity, there was already a difference of 2.5% between the models, rising to almost 10% difference at 30% porosity. And I had done the same for a C44. So I have, again, we can see that using our model, it's lower than the standard approach. And again, I've plotted the discrepancy. And again, you can see that at 30%, we're reaching up to 16, 17% discrepancy between the models. So what we came up with was some threshold porosities for when each model should be used. So these are just three particular elastic parameters that I'm showing in this presentation. In the paper, we tested various other ones. So we found that if your porosity is in your material, your biological tissue sample, your whatever material it is you're looking at, is less than 2%, then technically either model is going to give you basically the same result. If you go beyond 2% though, you're much better off to use our new model, because that's the one that will predict the behaviour more accurately. And similarly for E3, you get a bit more leeway, you get up to 8% porosity, and with C44, you get up to 5% porosity before our model takes over and provides a better and more realistic representation of your tissue. So then I want to mention how we extended this work then to a sort of first approximation to heart tissue modelling. So before I speak about the model, I want to just give a bit of an overview on heart modelling, because I'm not sure the background that people have on myocardial infarction or heart modelling. So the heart obviously is a muscular organ. It has 4 chambers and a thick muscular wall that contracts to pump blood around the body. So the heart wall itself has 3 different layers, and the most dominant layer is the middle layer, and is known as the myocardium. The myocardium is made up of extracellular matrix, elastic myocyte cells, and variety of collagen, elastin, and blood vessels. And those blood vessels are responsible for bringing the blood to the actual myocytes themselves. These muscle cells need oxygen and nutrients in order to allow them to keep contracting. Now, obviously, this is just looking at the heart in a structural approach. The myocytes are joined end to end at things called intercalated discs, and these are related. So these are where the electric currents are conducting from myocyte to myocyte, because an electric potential develops through the heart, causing the myocytes to contract and relax, and that's what triggers the heartbeat and how it pumps blood around the body. So the structural features in the heart are very, very important for its function and also for that electrical conductivity. If the heart becomes damaged, it will not conduct the electrical current as well, which will result in a less efficient heartbeat. And also, if the heart is damaged structurally itself, it also will not beat as efficiently. So it's a kind of two sort of problems that are arising if you have damage to the myocytes or any of the surrounding ECM. So what I want to look at here is I want to look at the case where a myocardial infarction has occurred or a heart attack. So in that case, the myocytes are supplied by these sort of surrounding vessels. The vessels become narrowed or blocked, and they stop enough oxygen reaching those myocytes. The myocytes become necrotic and damaged and begin to die. You cannot have the heart with sections of damaged dead tissue. For homeostasis reasons, the heart has to then produce a thick collagen-rich scar that bridges over that gap where you've had the myocytes die, and it does this in a way to retain the structural integrity. However, the structural integrity is there now with that scar, but its efficiency is now damaged because the heart cannot contract as well with this very stiff piece of tissue that's in its muscular wall now. So what the heart tries to do itself, and what medical advances are trying to do, is it tries to it increases the volume of the myocytes that are surrounding that damaged infarcted zone in an area called the border zone. And by swelling up those surrounding cells, you try to improve the elastic compliance of that scar tissue. So that's the sort of modelling scenario that we are going to be trying to approach with our different geometries and our different models that we've come up with. So here is a very microscopic zoomed in section of the myocardium. And again, I've taken my approach that I look at a tissue under the microscope and I try to create a sort of very simple but key representation of what I'm seeing there. So I am seeing quite a bit of extracellular matrix tissue. So that's what this dark blue colour is here. And I'm seeing these bundles of elastic myocytes. So I've just put this in just as a light blue circle because it's got a sort of circular rearrangement. Then I'm seeing blood vessels, and in this case, I've just put in some channels. In future works, more recent ones I've published, we actually include these as blood vessels rather than channels, where you have different conditions about the vessels perfusing the poroelastic material. So this is my structure, and I'm assuming that this is again, very basic, but a very simple structure for the healthy myocardium. So I have an intact bundle, an intact compliant ECM, intact compliant myocytes, and nice channels. In my diseased case, I have created something where some of the myocytes have unfortunately become too damaged beyond repair, and therefore have had to be replaced by the ECM stiffening into a hard collagen-rich scar. So that's what I have here. I still have my vessels, which we have assumed still have the correct arrangement, but we can shrink their diameter to simulate the underperfusion when the myocardial infarction is occurring, and an overperfusion on overstimulation of drug treatment to try to reperfuse the myocardium tissue. So what we are going to do now is compare the difference between a healthy and diseased simulations using these 3D geometries. So in this here I have some plots now. So I've just again randomly picked a couple of the elastic parameters that we solved in the paper, and we're going to compare those. So I've compared my C44 shear for my diseased, which is this much higher up, and my healthy case here. So as we can see, the C44 is a much lower value for the shear in the healthy case, and also its decrease with the increasing porosity is much smaller than our disease case. So again, just to explain the modelling scenario, the reason I was mapping this to an increased porosity was to look at the perfusion of the tissue. So to go from when there's almost no fluid reaching through to when there's far too much fluid reaching the tissue. And again, we did the same with the Young's modulus E1. So this was the one going across the situation when the fibers are coming down like this and we're pulling the material in that direction. And in fact, the infarcted myocardium has a much higher E1 than the healthy case. It's in fact almost double the stiffness. And indeed, the stiffness of the infarcted case does reduce dramatically with increasing porosity. So that goes towards the ideas that increased reperfusion of the heart muscle post myocardial infarction is very good at softening the heart and increasing its compliance. But we do need to think about which levels then become detrimental to the efficiency of the heart. So I've also presented here, this is a slightly different analysis. So I'm looking in this time at C66, which is just one of my other shears, and my E3, which is my other Young's moduli. And you'll see here that I have different lines for each of the different ones. Well, each of these coloured lines represent a perfusion level of the tissue. So I've got about 5% through to 20% perfusion. So that's the vessel volumes. And I've this time carried out my analysis in terms of increase in myocyte volume fraction, and that was to replicate the swelling of the myocytes in the border zone to try to again improve the compliance of the heart muscle itself. So both of them, as we can see, are decreasing with the increasing myocyte volume, and the behaviour is consistent across the 4 fixed fluid volume fractions. So we see that when we have the smallest perfusion, we're still getting the stiffest result, through to when we have the highest perfusion, we are getting the sort of softest result. So again, that tells us that the reperfusion is a good thing, but also that this homeostasis mechanism of the swelling of the surrounding myocytes is also doing a good job in bringing back that material compliance. So typically, the healthy shear for the myocardium is approximately 10 kilopascals, which is much lower than the sort of 24 that we're seeing for the infarcted setup. So this is, say, that 24 is probably somewhere about here. So this is when the perfusion is very low. So that's when the vessels have become very constricted and the tissue is, the myocytes have died, the scar tissue is coming in. And then, but obviously with that swelling of the surrounding ones, we are bringing it down if we remain at a 5% perfusion. But by increasing our perfusion, we can bring it down until, in fact, with the bulk 20% perfusion, we can almost return to that typical healthy shear that the heart would like to have in those myocytes. So just to provide you with a few conclusions to this work, so we can say that for a poroelastic composite, so that was my new work, my new model, with porosities exceeding 5%, then the discrepancy between my new model and a standard poroelastic approach, when computing the different Young's moduli and the shear, if that porosity exceeds 2%, if the porosity exceeds 5% and then the discrepancy between the model exceeds 2%, then we should use my model rather than the standard poroelastic approach. For materials with less than 5% porosity, either model setup can produce approximately the same results. And in fact, we have proved that our new model should be considered as a valid alternative to a standard poroelastic approach when modelling materials that have an inhomogeneous microstructure. And that was the main point of those different works, was really to show that it does make such a difference to consider all the elastic phases at the same scale as the pores, if that is what you see in your tissue sample. And in fact, this model, as I hope I've shown slightly with that extension to the myocardial modelling, it can be extended to much more complex geometries. We can have many additional elastic fibres, we can include different fibre angles, and fluid flows in a variety of different directions. So thank you very much for your attention, and I'd be very happy to answer any questions that you have.
Thank you very much, Laura. That was really great. Has anyone got any questions that they want to jump in with? Ellen Luckins has jumped right in with a question there, so I think you should be able to unmute yourself.
Hi, Laura. Thanks very much. That was really interesting. Can you hear me okay? Is that all right?
Yeah.
Great. So I was really interested to hear about your homogenization work with the poroelasticity. I wondered if you had to make any assumptions at all about the size of your elasticities in order to do the analysis. Like, did they have to be the same size to an order of epsilon, for instance?
I think in this case we didn't need to. With the solid parts, there wasn't any need to scale things differently. With some of the other works that I've done with perfusion and things, sometimes you have different epsilon scalings for the fluid compartments based on if it's the flowing vessels maybe needs an extra epsilon squared or 1 over epsilon if you're looking at the different sort of fluid slip conditions in vessels with porous media. But in this case, we did carry out a non-dimensionalization of all the fluid structure interaction problem before the upscaling. We had the classical epsilon squared in our fluid stress to account for the flow in a cylinder. But other than that, the elastic parts, no, they didn't need any different scalings.
Wow, that's really interesting. I guess especially for your biological applications, you'd expect things to have similar sorts of scales, but maybe if you had like a really rigid scaffold with something really squishy on it as a very different elasticity you'd get something else, but maybe that's not relevant.
No, definitely. I think that in this case, things were, even though we were saying that one phase was maybe much stiffer because of disease or whatever, they were still on the same order of magnitude of sort of stiffnesses. They were all, say, all on kilopascals, even if one was, say, like 1 through to 100, but it was still that sort of small scale changes.
Cool. Nice. Thanks very much.
Thank you.
Yeah, Tom, I can see you're next, I think.
Yeah, so about the homogenization process again. You mentioned that, I mean, you have this unit cell approach and you enforce periodicity strictly. Would it be possible to sort of relax that assumption somewhat if you zoom out enough? As it were. So would it be okay to have something that is basically periodic or at least evenly distributed and then apply some sort of mean field argument?
Yeah, I mean, there's definitely an awful lot of those things that can definitely be done. This was done with the sort of the strictest conditions, say, but you don't even have to have that periodicity per se. You can have just local boundedness of fields, but then you're not going to get things that can be solved just on periodic cells. You would have to solve across the whole microstructure, which is which creates a lot more computational problems. What you can also do though is, as you were saying, you can have one section maybe of the material with a certain microstructure, you can have another section with a slightly different microstructure, and then you can bring those things together at, say, a higher up scale. In a work I'm working on at the moment, it's sort of 3 or 4 different underlying periodic cells you can see, but they are varying across the tissue. So it wouldn't have been realistic to say that the one cell was capturing that whole tissue. So if you can divide into somewhat semi-repeating blocks, there is always something you can do to create some sort of homogenized model. And again, that this was assuming the macroscopic uniformity, where you were seeing the same underlying microstructure at every macroscopic point. But I know there are various people approaching these things a bit differently as well. So there's quite a lot can be extended on that front for certain tissues or materials.
Ayush was next. I have more questions.
I think Ayush is showing us next, so I'll hand over to him next.
Yeah, so my question is, why in the governing equation, why you use the Darcy's law, why not the Darcy's Brinkman equation? Because as far as my knowledge is concerned, Darcy's law is much inaccurate near the interface between the porous and free flow regions.
So what we actually obtain the Darcy's flow as an upscaling, because we are just assuming a very simple Newtonian fluid flowing in the pores. But I do agree that in certain porous structures and porous materials, Darcy-Brinkman would be more realistic to certain relevant scenarios. Again, there's so many things can be extended to the work that I've presented today, but no, you're completely correct for certain things that we would be able to start from a slightly different starting point even to arrive at Darcy-Brinkman via the upscalings. But this was just the simplest possible.
And another thing is that most of your results are based on the variation with the porosity. Why not with the permeability?
Well, in this case, these are just the solid simulations. So when we do homogenization, the simulations are drained. So we've removed the fluid from those cubes. And so it's just really based on those holes that are there. If we were to consider if I was to present to you the simulations for the hydraulic conductivity tensor, in that case, we maybe could consider things in a sort of permeability, or if we were looking at cases where I've added vasculature via proper vessel-based equations, we could show the influences of permeability maybe a bit more strongly there.
Yeah, thank you from my side.
when you do your scaling up based on your unit cells, you end up with essentially a bulk equation. It would be perfectly valid to put that, to simulate that bulk equation on a finite domain. But of course, we're a perfectly reasonable thing to try, I suppose. But of course, the assumptions of how that would be derived are not valid at the domain boundaries or near the domain boundaries because your unit cells go wrong. Is there a way around that? Asking for a friend, because that would be very useful. Have you thought about that at all?
It's admittedly not something that I've really thought too much about. I mean, we always say when we're deriving the model, we don't need to focus too much on what the sort of external boundary conditions would be because they don't make any change to the upscaling per se. But I'm not confident in saying we can. I would need to think about what exactly could be done at those, because if you've got so I'll give it some thought. I'm not completely sure what we could impose to try to overcome that at those boundaries.
I guess a first sensible thing to do would be to look at a semi-infinite, a semi-periodic domain, right, and see if you could come up with something there. But I think Raimundo and Ellen both have ideas here.
Some bits of work on this. I've worked on some problems like that myself, but maybe we should chat another time, Tom. But there's some groups in France doing stuff like that as well. So I can send you some papers if you're interested.
Raimondo's also got his hand up, so I don't know if you want to...
just great. It's always a pleasure to listen to these things. I mean, so just a couple of comments. So when it comes to the Darcy-Brinkmann, basically, someone asked, so basically you have to go on at the second order. So if you address the interplay between a Newtonian fluid and an elastic matrix, and then you go on at the first order, you get Darcy, then you get Darcy again, but a modified version of Darcy, and then again, if you go on at the second order epsilon square, eventually you close the problem, you get Darcy-Brinkmann, which means that Darcy-Brinkmann should be used when the pores are not as tiny as in the case of Darcy. Otherwise, so and that would be a more accurate approximation. So that's the point. So if you have that the scale separation is appropriate but not quite as you thought it should be, then and there are papers of a French school in the 90s that say that. Also to pick up on the remark on the failing of a periodicity assumption on the boundaries, the solving that's a very it's a niche but not so niche kind of observation. I kind of looked into that years ago, and I'm aware of some work by Bernhard Schläfer, is now emeritus in Padua in the 90s, and he was using some sort of exponential function to sort of provide some sort of analytic continuation with solution at that boundary or something. So that's something definitely that one could look into. So these are theoretical issues I've been looking for some time. I mean, so please, if someone has some papers, that's also interesting for me, because this is something that just a few people focus on, but they can be relevant in some specific specific situation. So that's and on the Darcy-Brinkmann, I worked recently also on the Darcy-Brinkmann. That's true There are some situations where this can prove as being more accurate. Here, I suppose we were happy with the leading order, result, and the leading order. You do get Darcy's law. That is what it is. So one has to go on and admit a poorer scale separation. And then eventually you'd find that. So basically, Darcy-Brinkman is always more correct, but if the scale separation is very much pronounced, then in theory you can content yourself with Darcy's law. That will be my 5 cents to add to the discussion, if that makes sense.
Right. Oh, there we go. Ellen has started sending some of these papers across. So I was
You're
going to
so lucky.
say.
There's a group of folks in France and Italy, I think, that have started looking at this a little bit for thin membranes to think about effective boundary conditions across them. Anyway, sorry, you were going to say more things, Joe. I'll keep putting some things in the chat.
I wasn't actually going to say more things. So carry on if there's anything else that people want to discuss off of the back of that.
Well, that's you've interested me now, Tom, with that question. We need to think about that.
Should leave it for a little bit longer. Are there any other papers, Ellen, that you're aware of?
I'm just trying to find things. So yeah, there's this group that have started to think about flow across a membrane. So that's not poroelastic, it's just flow, and it's a thin membrane. So it's a jump condition they're deriving across it. But they use a combined boundary layer and homogenization technique, which is something I've also been doing for free boundary problems in porous media. So I can find that one. And there's some people that have kind of derived Beavers and Joseph carefully with a similar sort of method. But there's just small pockets of it. I agree with you, Raimondo. There's little bits here and there, and I hadn't heard of the one that you had, so that's interesting. I'd be keen to see that.
The Beavers and Joseph is another kind of worm, because it's very fascinating condition where there's half of a community that hates it, half of a community that loves it. So I always try to cite both and say that I just want to use it because it's convenient, but I don't want to enter into fights. I mean, I don't know if you guys are aware, but some people of a French school were fighting with each other somehow. So and I found that they are kind of sending each other letters. I found that recently, because one has to be careful and cite everybody, so they're happy, because otherwise these people are, there's something more delicate than you want it to be, it seems. I mean, I actually know too much of an, I used that in a couple of papers, and it's actually a very general condition, because it contains basically both no sleep and free sleep as particular cases, so that's why I would use it. But people say, okay, once you go to 3D, then you cannot use it. Oh, but I guess you can. It's kind of a mess. So that's another kind of worm, I think.
Great. Well, I think that did spark some good discussion that hopefully might be able to continue. I will, when I save the recording of this, I will copy the contents of the chat as well, so that everyone will have access to everything that's been shared in the chat when they come to view the recorded version of this seminar. But unless anyone has got anything else they'd like to add, I think we might leave it there for this month's seminar, and thank Laura again for a really excellent talk and some great discussion that came off of it. So thank you very much for agreeing to speak to all of us today.
Thank you very much for inviting me. Thank you.
Thanks a lot.